Wayne, you're upside down.
Well, I was reading, Trev.
Oh.
We're still on topic 2, genes and health. We're going to do topic 2.2 in this part 1 and part 2. Why is CF mucus so sticky?
So Wayne, why is CF mucus so sticky?
Good question, Trev. The reason that CF mucus is so sticky all revolves around the point that people with cystic fibrosis can't make a particular protein, the CFTR protein up here.
Very nice. But you have to know a bit more about proteins.
Yes, to really understand this you need to know a lot more about proteins. You should be familiar with the idea that it's all made up from amino acids.
Yeah, and loads of them make a protein. So we're going to go through the structure of, or basic structure of an amino acid. In the middle we have this bit, which is the carbon bonding to a hydrogen and an R group.
R stands for residual, and that changes with the different amino acids.
It does. It can be lots of things. It can be hydrogen, most simple, and it can be a lot more complicated.
You don't have to know all the amino acids, you just need to appreciate that this can change.
Yeah, and keep making different things.
You've also got to know that it bonds to an amine group and a carboxylic acid group, and you need to know what that looks like.
Yep.
Oh, red. I like
Right,
it.
let's do it. So in the book they write it this way, so write it this way as well.
I'm doing the amine. Trev's doing the carboxylic acid.
Okay.
And that is the structure of an amino acid.
It is. Now this changes when it gets into a chain.
It does. Now, for this, because it's made up of lots of amino acids, this must bond to more of this.
It does.
And you should be familiar from topic 1. This OH and this hydrogen are removed so that this carbon will bond to the next nitrogen, which will bond to the next. You'll notice this will produce H2O, so what kind of reaction is that, Trev?
I think it's a condensation reaction, Wayne.
Just like in topic 1.
Alright.
So you need to be familiar with that, which leads us on to the structures of protein.
Yeah, there is quite a few.
There is.
They're set into 4 sections, but really I guess there's sort of 5.
Yeah, because they split up again after it.
Yeah. So, beginning, there is primary, which is just a straight chain.
Yep, so it just does this over and over, nothing more, and that's primary.
That's primary. So primary is just a straight chain. Then, secondary, this is the one where there's 2 types. There is alpha helix.
And a beta plated sheet.
Yes. Now, these are formed... These are different because of the way the hydrogen bonds form.
Yes.
So in one they form as a way to make it hold a helix shape, and the other one they sort of form across, I guess where you'd say the folds in the sheet would be. And the diagrams of that are in the book, you just have to understand it.
Yep, so a bit like DNA, the hydrogen bonds are between the opposite strands of this helix.
Yep.
In the beta plated sheets, the hydrogen bonds are between the layers. And the hydrogen bond forms between this O and this, well, this H, because this one's been removed, but
yeah.
And it bonds between there, that's where the hydrogen bond is.
Okay, the next is tertiary structures. This gets a bit more exciting, there's more types of bonding that can happen here. If your residual group happens to have sulfur in it, also forms sulfide bonds. And it gets a bit more of a complicated shape, it sort of all goes wibbly wobbly, timey wimey. Yeah.
But again, it will create these hydrogen bonds still in this one. This just has more.
It's sort of like adding them all together.
It also creates ionic bonds if it has the right R groups.
It does.
By van der Waals forces and stuff like that also come into play, but you don't need to know about that.
Yeah, you can see by that the R group's going to change the structure a lot, and that's why they're different amino acids and different proteins.
Yes, it is. So that's just one straight chain doing all this wibbly wobbly timey wimey stuff. There's also quaternary structure.
When you have another one and you put them together and they go that's quaternary, just like a really complicated version of tertiary.
Yes, just more than one chain.
Just more than one chain.
So these are the main types of structure for the proteins.
There it is. And then it goes into 2 more sort of groups that they can all be part of.
Yes, in red you have globular.
You do, and fibrous.
Globular compacts it all with all of its bonds into a sort of spherical shape or an oval shape or something along that. As 3D image of something.
A bit like a blob.
A bit like a blob. That's how I think of it. Glob. And fibrous.
Which is more sort of like fibres in like a string or something.
Yes.
Twisted line.
Examples of this is stuff like red blood cells and stuff like that. You can see they're roughly round. They're not quite round. You should know. But examples of that. Fibrous stuff like your hair, the keratin protein. Because it's nice and straight. It's nice and simple.
Not born. So, yeah.
Yeah. And that's these are the further divisions of these
all
divisions.
different types of protein. I think that's pretty much
That pretty much sums
protein.
up proteins.
And part 1 of the video?
Not quite true.
Not quite. Oh.
We have some further stuff to do if we get to the right page.
Okay.
Next we go on to cell membrane structure.
Oh.
So
Alright.
let's crack on with that.
Let's crack on with that.
We're back
Hello.
with cell membrane structure, the phospholipid bilayer, and then the fluid mosaic model.
But first, what's a phospholipid?
Has the word lipid in it, which you should be familiar with, so it's not that scary. I'll draw it up.
No, but you do need to know it. In Unit 1 we spoke about lipids, and they're basically fats and the structure of them. So, it's got a glycerol in it, and then it's got a fatty acid bit. Now if you do chemistry, just remember the first bit really well. It has these, the skeletal bit, which denotes a carbon chain. Only problem is, it's got a phospho bit.
Yeah, there's no way to fit a phospho on this at the minute. So what it has to do is actually replace one of these fatty acid
It
chains.
does. So we lose all this.
All the way up to here. That all goes. I'm actually going to flip this O and this H around, because that's how it's done in the book. The O is that side, the H is that side.
It is.
Carbon can make 4 bonds, so that's another
Another
one.
hydrogen. And then the phosphate joins in. So there's the phosphorus,
Double
like that,
oxygen.
oxygen, left, oxygen, hydrogen, oh an X, sorry.
It
That
can
could be a hydrogen.
The X
but-
can change, you don't have to worry about that at all.
No, but it can change. It's like the R group.
Yes. You should realise that makes this slightly negative and this all slightly positive.
This gives phospholipids a special property.
Yes, it makes it hydrophilic and hydrophobic, with this end being hydrophilic because it's negative, this end being hydrophobic, water hating, because it's positive.
Yeah, you should remember that from GCSE. Philic loving, phobic hating.
You don't have to draw this out every time, that would be bizarre. There's an easy way of drawing it, which
There
Triv
is.
is now going to do.
It's a little circle which denotes the top end and sort of like the skeletal things, like that.
So if I've got a blue pen that I can use, yep.
Yeah, okay.
If we do a bit of water,
So we have some water.
so that.
A few ways things could happen. It would naturally, if you just put it on top, it would go like that. It would just sort of hang. But if it was actually put into the water, quite a lot of them would sort of dead join together.
Yes, because this part wants to be out of the water, but it can't just detach itself.
No.
So the result looks something like this. Get your own little circle. It's really hard to draw on a board. And then all of the lines go in, so they've effectively got a water-loving shield around them, which would produce sort of like a bubble in there of no water.
It would.
These would actually be under the water. They would be mostly submerged up to here.
Yeah.
But that's just to show this part loves the water and that part hates it. So that pretty much sums up the phospholipids, but phospholipid bilayer.
That. I have a bicycle that's got 2 wheels.
Yes, bi means 2, so you'd have something to do with 2 of these. In fact, you have many of them. It gives them a double layer, that's what it means.
It does, and that's how a membrane forms. It sort of has a bit in the middle where things can go through.
Which leads us on to the fluid mosaic model. That's what that's all about. So let's rub all this off.
Okay.
Now, the fluid mosaic model is something you should be familiar with, and it does come up a lot in
It
your
does.
Unit 1 exam.
So we're here to talk about how to prove it as well, which is what, well, this bit is about.
Yes. So, because it's a bilayer and it's a phospholipid bilayer, you can imagine it's the phospholipids that make up the 2 layers.
Yeah.
So we've got, I'm not going to draw out a massive one, I'm just going to sort of represent, there's a few
Rough drawing of that.
This. So on and down. Got a few more on the other side.
It does take quite a long time to draw.
It's
Then-
normally drawn for you.
Yeah, in the
just have to
exam, you
annotate.
sort of had to draw what ends hydrophilic, what ends hydrophobic, what it is, and label stuff. Yeah.
Yes. And you should notice that effectively makes it waterproof, nothing can get in and out. So how
Yeah.
does stuff get in and out?
Well, there's a channel protein, and this starts filling out what a membrane actually looks like. So we have this sort of like a tube, sort of like a membrane is, and in the middle we have a channel protein. This lets things through. We'll go on about that more
Arrow here.
detail later, because that's actually the bit that CF affects.
Yes.
And then there's bits sticking off.
Yep, you've got glycoproteins and stuff and sort of.
Glycoproteins just sort of come off of it. And this
I'm drawing it like this. It's all a very rough drawing. This would continue on the other side a little bit. This
roughly
phospholipid
looks like
bilayer.
that.
So stuff gets in and out through here. The proteins control what gets in and out, and
They
like
do.
Triv said, there's A little bit into the future of the book, that's what CF is all about. That's part 2 of this video, we go on to that.
We do.
And the CFTR protein in particular. This actually sums up our part 1 of this video. We've done all of this. Next in the book is diffusion. We start that in part 2.
Okay, so sorry about that.
And we don't sum up in this video, we're going to sum up in part 2, we're going to sum up this and the next video.
Okay.
Let's go get cookies.
Cookies.
Again.
Cookies.